Welcome to this PHA Live webinar. We will get started in one moment. Before we get started, we will be broadcasting this webinar in both Spanish and in English. So if you would like to listen in, we have the instructions here on the screen. Please click the globe at the bottom of your screen and select the audio in which you would like to hear. That will translate the information. If you would like to hear English, you will hear the panel. And if you would like to hear Spanish, you will hear our interpreters. Again, welcome and thank you for joining us today for this PHA live webinar, Hope Through Research in Pulmonary Hypertension, Emerging Developments. We're so excited to have you here today for this very new thing that we're doing with PHA Live. We're able to offer interpretation and simultaneously in Spanish for this important information. And also to bring this to you, we would like to thank our sponsors for this webinar series. We would not be able to have this interpretation and the translation without our wonderful sponsors sponsoring this PHA Live webinar. One of the things that is important for PHA to spread and to continue its mission is to be able to provide educational resources like this one, like a webinar, like our PHA classroom and other resources we have available. By becoming a member, you help us to continue that message and that mission to hopefully, raise awareness for PH, find a cure, and empower our patient and community to have more information about PH. Also, as a professional member, you are able to receive discounted admissions, sorry about that, to PHA International PH Conference, free CME credits, and so much more. And right now, we are running a special rate for PHA membership for professional members. So if you are interested and would like to receive more information, please feel free to visit us online or join using our information here below. You're able to scan the code with your phone. Some additional information before we get started, just a couple of housekeeping things. So we will have a Q&A immediately following the PHA Live webinar. You can use the Q&A button at the bottom of your screen to be able to submit your questions to the panelists. And then after the webinar, there will be a survey that will pop up in the browser for you to answer. So please complete that survey because it helps us to know exactly what you are interested in learning about and helps us to plan for the next year. So again, welcome and thank you for joining us for this PHA Live webinar, which PHA is doing in partnership with the Foundation Against Pulmonary Hypertension in Spain. So we're so excited to partner with FCHP with this webinar and to have our lovely panelists here who will introduce themselves now.
Good afternoon. My name is Julia Feitner, and I'm a PH patient. I was diagnosed 15 years ago when I was 34 weeks pregnant with our second child. And at that point, I was sick enough that within 24 hours, I was intubated. He was born via emergency C-section, and then I started on Flolan. It has been a long uphill battle since then. And I am here today in huge part due to two of the clinical drug trials I've had a privilege to be in, the imatinib and currently sotatercept.
My name is Catherine Simpson. I'm an assistant professor of medicine at Johns Hopkins, and I am a PH provider.
My name's Anna Hemnes. I am a professor of medicine at Vanderbilt University Medical Center. I'm a pulmonologist and I do research in pulmonary hypertension and also take care of patients with pulmonary hypertension.
And my name is Matt Lammi. I'm an associate professor of medicine, pulmonologist at Johns Hopkins University, and I have the pleasure of chairing the PHA's research committee.
My name is Eric Austin. I'm associate professor of pediatrics at Vanderbilt and the PH program director here for children. I'll be active on the next webinar, but wanted to say hello and be in the background today. Thank you.
All right, great. So we'll get started with the presentation. Again, I said before, I'm the chair of the research committee. I have the great fortune to be partnering with a lot of wonderful investigators throughout the country. Eric Austin is on our committee, and it's really a shared committee of pulmonary cardiology, pediatrics, and adult researchers, and it spans the investigations from basic laboratory science all the way to patient-centered research. I'm just going to give you some perspectives on PHA's commitment to research. Currently, there are 3 research grants funded by the Pulmonary Hypertension Association, which I'll talk about in turn. But the 3 are Innovation in Pulmonary Hypertension Research Award, Pediatric Research Award, and an Early Career Mentored Scientist Award. Firstly, the Innovation in PH Research Award. Its intention is to fund new areas of research that may not be possible without PHA's support. So these are really targeting projects that are very innovative and a little bit high risk in the fact that they're really trying to push the field forward towards that cure that PHA is trying to support. The grant is for up to $60,000 per year for 1 to 2 years of research. And we were fortunate enough through the support of PHA to have 2 winners of this award through this last 2023 cycle. The first winner is Kei Yuan from Boston Children's Hospital, whose project is perivascular cells contribute to flow-induced pulmonary hypertension. So this is really looking at an innovative and new way of thinking about a particular cell along the pulmonary blood vessels that can contribute to pulmonary hypertension. The second award went to Adam Andruska from Stanford Medicine, whose project was T-cell vascular signaling as a therapeutic target in pulmonary arterial hypertension. So really looking at the immune system and how that may be a target for treatment, really trying to push development of new therapeutics for pulmonary hypertension. Pediatric Research Award. This is a funding mechanism that addresses unmet needs in pediatric pulmonary hypertension research. So again, PHA is supporting research from basically birth all the way through the lifespan. This is up to $65,000 for 1 year. And our most recent winner is Paul Christer, who's at Cincinnati Children's Hospital Medical Center, who's looking at pulmonary vascular development in bronchopulmonary dysplasia-associated pulmonary hypertension. So this is a very challenging condition to treat in newborns, and Dr. Christer's work is really trying to move that field forward. The next award is the Early Career Mentored Scientist Award, and this funds projects that are being done by young junior pulmonary hypertension researchers who have applied for funding from the National Institutes of Health, but didn't quite get funded. So PHA is providing $65,000 for 1 year to allow that applicant to generate more information and more data so that they can be successful in their next application. Our most current awardee is Rupa Sadaya, who's from Penn State University, also looking at bronchopulmonary dysplasia. So again, another pediatric award. And this is looking at predictors of pulmonary hypertension in this condition. And lastly, this is an award that its final cycle was 2022. This is the Aldrighetti Research Award for Young Investigators. And this is a 6-year total grant commitment, which was sponsored by Janssen in partnership with PHA. And Dr. Catherine Simpson, who will be speaking to you about our project shortly, was looking at compartment-specific kynurenine pathway metabolism and PAH, and she'll give you a lot more information about that and how this award and the PHA support has been important to her career development. Outside of these important research awards, PHA has other activities, including the Pulmonary Hypertension Association Registry. This is the largest active US-based registry of patients with pulmonary hypertension. This is a rich resource for research, especially by young investigators, and there's been 17 publications so far, ranging from topics such as the impact of COVID-19, regional variations in pulmonary hypertension, quality of life, as well as social determinants of health in PH. Also, we have the PVD-omics project, which Dr. Hemnes will be talking about, but this is a partnership between PHA and the National Institutes of Health, which really is rethinking the way that we classify patients in order to improve outcomes, and more to come on that a little bit later in our webinar. And the last thing I want to touch on is the PHA Research Room. This occurs every other year at the PHA International PH Conference, and it's an opportunity for individuals with pulmonary hypertension to volunteer as research subjects at the conference itself, to provide blood samples, questionnaires, other simple metrics that is very helpful for researchers to really understand the patient perspective on pulmonary hypertension. In past conferences, over 200 people have volunteered to take part in the PHA Research Room, and this is, again, demonstrating the strong commitment to research that the Pulmonary Hypertension Association is invested in. And with that, I will turn the mic over to Dr. Simpson, who's going to talk about her research that has been funded by the Pulmonary Hypertension Association.
Thank you so much, Matt. So I'll tell you all a little bit about my research currently in progress, a substantial portion of which is funded by the Pulmonary Hypertension Association. As I said earlier, my name is Catherine Simpson, and I'm also at Johns Hopkins University. So the notion that frames much of what I'll tell you about this afternoon is that we need more refined approaches to managing pulmonary hypertension. And I use the word managing in particular because I think that this encompasses more than just treating PH. So I'll give you some examples of work that we've done to try to improve upon phenotyping, so understanding the fundamental basis of why particular patients presented the way that they do, assessing response to therapy, so namely trying to develop biomarkers of pulmonary hypertension, and also trying to develop precision treatments that probably will be given in addition to the currently available pulmonary vasodilator therapies. The lens that my work uses to work on these topics is something that the field has called omics in the last decade or so. Most of you are probably familiar with the genome. All of us have DNA, which provides the genetic code for all of us. But that's actually fairly far removed from phenotype or the clinical expression of that DNA, and there are lots of steps along the way. So the DNA that we have is transcribed into mRNA, which has been translated into proteins, and then those proteins all work in the body to enact various processes. And the sum total of that process or those processes are what we think of as metabolism or the metabolome. And a lot of the work that I'll show you today concerns metabolomics, which is many would say the omics that is closest to the clinical expression of phenotype in our patients with pulmonary hypertension. So first, I'll start by showing you an example of metabolomics used for more refined phenotyping. So what this is, what you're looking at on your screen, is a survival plot of a cluster analysis that Dr. Hemnes, myself, and others in the PVD-omics network performed in that cohort. And so what you see here are different colors that are representing different clusters that were arrived at by modeling only the metabolism, the metabolites alone of patients with pulmonary hypertension. And so when we did this modeling approach, we found that there were 3 clusters of patients, and each cluster had metabolism that was very much in common and very different from other clusters of patients with specific forms of metabolism. And you can see in the survival plot that these different clusters of patients experience different survival periods. So the patients in the red cluster experience the best survival, and the patients in the gray cluster experience sort of the worst survival, poorest survival over time. And again, these cluster assignments were made just on the basis of metabolite information, just molecular information. When we looked at clinical information, we found that things like PAH subtypes, whether a patient had idiopathic PAH or connective tissue disease-associated PAH, those things were balanced across these clusters. And then furthermore, when we looked at common clinical characteristics that we care about, like right heart catheterization measurements, such as right atrial pressure, pulmonary vascular resistance, some echocardiographic measures of right ventricular function, these measures also differed according to cluster assignment. And so here is an example from our research of how we might use molecular information such as metabolites to better phenotype patients and understand a little bit more about their fundamental biology, and potentially in the future tailor treatments to that. The next thing I'll show you is just an example of some biomarker work that we've done. And one day we think maybe we can use better biomarkers to help assess response to therapy once patients begin on pH-specific therapies. And so this is an article that we published looking at metabolic profiles as biomarkers of hemodynamics in patients before and after a bout of exercise. And here's just the main figure to highlight from that paper. And it's not immediately intuitive. And so just to orient you to it, each column here, moving from left to right, represents a biomarker. So the leftmost biomarker is something that you may be familiar with, proBNP, our current clinical gold standard biomarker for pulmonary hypertension. And the rows represent different hemodynamics, different clinical measurements that we care about. And this R squared statistic that's in each box, you can think of as the amount of variability in the clinical measurement that's explained by the biomarker. So you can see in the first column that proBNP explains 26% for rest mean pulmonary pressure, 30% for exercise, of that variability in the different patients in this particular cohort. By contrast, these pre-exercise metabolites, so these are various combinations that are modeled, selected, of maybe a dozen metabolites or so, that were much better at explaining the variability that was present in pulmonary artery pressure. So 81% of resting mean PA pressure was explained by this pre-exercise metabolite profile. And then just to be very concrete, the pre-metabolite profile was able to explain 93% of the variability in exercise mean PA pressure. So, we think that with further rigorous study and careful study, we can potentially in the future come up with better biomarkers that report on disease and disease activity in real time over the course of disease for patients. And the final example that I'll show you from the research that we've been doing, and this is work that's more directly or that is directly funded by the PHA, this is an example of trying to understand early disease pathobiology to formulate precision treatments. And so this particular study is based in an observation of scleroderma patients that are presenting to their scleroderma doctors for routine clinical care. And this group of patients is followed over time, and they present every 6 months or so for routine pulmonary function testing and echocardiography to surveil for the development of pulmonary hypertension, which scleroderma patients are at higher risk for. And as they come and receive their routine clinical care, they contribute blood samples. And what this has allowed us to do is this has allowed us to analyze the blood samples of patients that did develop PH while they were under surveillance, compared to those that did not develop PH. And what this gives us a look at is just an early look at the evolution of metabolism and the molecular basis of disease for these patients as disease develops. And so here's just an overview of what we found. When we looked back in time prior to diagnosis, so about 2 years prior to PAH diagnosis in these patients, we found that there were some molecules in the blood that were already elevated in patients that would develop PH compared to those that did not or would not. And over time, as these patients move forward in time closer to diagnosis, these levels continue to rise. And these topmost levels that were very different in patients that would develop PH included kynurenine pathway metabolites. So these are byproducts of tryptophan metabolism via the kynurenine pathway, which is actually the main route of tryptophan metabolism in mammals. This kynurenine to tryptophan ratio, which you can see in the top right of the screen, so that's 50% higher in PAH patients at the time of diagnosis, that's actually a surrogate of the enzymatic activity for the rate-limiting step of kynurenine pathway metabolism. So this is, of interest to us because this is a metabolic change that's developing as these patients with scleroderma develop disease, and there is potentially a targetable enzymatic step that we could potentially act on. So this led us to work that we proposed to the PHA. And that was to integrate other omics, transcriptomics, to try to understand more about those enzymes. So what you see here at the top of the screen is the pathway schematized. And you can see that as tryptophan is converted to kynurenine, there are a variety of enzymes that are responsible for that step. One of those enzymes is TDO. And in the volcano plot at the bottom, which is a depiction of differentially expressed genes in PAH lungs compared to normal lungs, one of the most differentially expressed genes is TDO2, which is the gene that encodes TDO. And so this provides more evidence that in the organ that actually manifests disease, that this pathway is likely relevant and important to pulmonary hypertension development. What we've done further with PHA support is we've looked into animal models of pulmonary hypertension to try to understand if there's a model system that we can use to interrogate this pathway further. This is a study that we've recently completed that was funded by the PHA. In this study, we used our Sugen chronic hypoxia model of pulmonary hypertension, and we examined tissue and blood levels of kynurenine pathway metabolites and enzymes as these rats develop disease very early in the course. Actually, these changes that are depicted here are developing prior to pulmonary pressure elevations in these rats. And what you see in these orange plots here is basically that the TDO2 gene that I showed you in human PH lungs is rising in a stepwise fashion as these rats develop experimental pulmonary hypertension. So the sum total of all this is, we think that we've found a pathway that may be pathobiologically relevant to patients with pulmonary hypertension, and we think we have a model system to examine this in. And there are drugs that have already been developed that target this pathway that are currently being studied in cancers in early phase trials that could potentially be repurposed to pulmonary hypertension. And so these efforts are all part of what we hope might be development of a new precision treatment. So with that, I'll stop and just conclude and state again that metabolomics may be useful for refining our current approaches to managing pH, just one tool of many, of course. And some of the examples that I've shown you today are for identifying biomarkers, gaining a deeper understanding of pH pathobiology, and the hope would be to eventually identify potential therapeutic targets. And with that, I will turn it over to Dr. Hemnes.
Thank you so much, Dr. Simpson. That was a great presentation. I really enjoyed hearing about what you're doing. I'm going to try to share my screen. Actually, hold on. You see my slide, or do I need to swap screens?
We see your slides.
Okay, great, thanks. So my job today is to give an update on research in pulmonary hypertension. And this is one of my more favorite things to do. The themes of the research in pulmonary hypertension in 2023 that I think are really relevant are basic and translational science. And oftentimes when I've given a similar lecture to this, I've actually highlighted Dr. Simpson's work, but because she just presented it, I will not do that because she did it in much more thorough detail, but I'm going to focus on junior physicians and scientists in this field. I think a major move is in precision medicine approaches, including PVDomics and immunophenotypes. And then I'm going to talk a little bit about novel therapeutics that you can see listed there. So the first part of the basic and translational research is from a prior Aldugretti awardee, Dr. Vineet Agarwal, who's a mentee of mine. And he's been interested in TGF-beta receptor 3 and understanding how it could affect BMPR2 signaling in PAH. So we know that in pulmonary arterial hypertension, particularly in heritable pulmonary arterial hypertension, there is a high percentage of mutations in this gene called bone morphogenetic protein receptor type 2, or BMPR2. And even in people who don't have mutations, there's reduced expression. But it is one of a class of very similar receptors that sit on the cell surface. BMPR2 is one, TGF-beta receptor 3 is another, and there's others in this ALK pathway. And you can imagine that dysfunction in any of these may work together to cause pulmonary hypertension. So Vineet's been studying TGF-beta receptor 3. These are all in the TGF-beta receptor pathway, all each of these cell surface receptors. And as preliminary data for this Aldugretti Award, Vineet looked at TBR3 expression in both control rats, and this is expression in the lungs, and their controls shown in blue. And then in 2 different rat models of pulmonary hypertension, one is monocrotaline and the other is Sugen hypoxia, and they're really the standard rodent models. And he was able to show that there was, in these pulmonary hypertension models, reduced expression of this TGF-beta receptor 3. And then he used mouse models to look at what happened to right ventricular systolic pressure, which is a marker of pulmonary hypertension that we use a lot in mice. And in, not surprisingly, wild type mice, when we give them Sugen hypoxia, this mouse model of pulmonary hypertension, there's a rise in RV systolic pressure. You can see in the far left columns. And then in the TGF-beta receptor 3 partially mutant, so they have 1 mutant copy, 1 non-mutant copy. When they get Sugen hypoxia, they have an increase in RV systolic pressure. And then to a greater extent, when we completely knock it out in another different kind of rodent model and expose them to Sugen hypoxia, we can get an even more marked increase in right ventricular systolic pressure. So that was an interesting finding, that this other receptor in the TGF-beta pathway may play a role in pulmonary hypertension. And then Vineet went ahead and looked at how this may be relevant to humans with pulmonary arterial hypertension. And the first thing that he did is looked at lungs of patients who have pulmonary arterial hypertension who have gone on for transplant. And we took the explanted lungs and were able to show that compared to normal controls, there was reduced expression of this TGF-beta receptor type 3. And then lastly, he found a single nucleotide variant, so like a gene polymorphism that was associated with higher or lower TBR3, TGF-beta receptor 3 expression. And in the folks that were expected and predicted to have lower TGF-beta receptor 3 expression, they actually had, in a large data set, a higher PA pressure on right heart catheterization, again suggesting that there may be a role to play for this different TGF-beta receptor in causing pulmonary hypertension. So the second main theme that I was going to touch on was PBD-omics, and Dr. Simpson talked about this initially, but I'll expand on it. So this was a multi-center study that was funded by the NIH and also by the Pulmonary Hypertension Association. And the goal was to perform comprehensive phenotyping and endotyping across all kinds of pulmonary hypertension. So not just pulmonary arterial hypertension, but could be due to lung disease, left heart disease, chronic thromboembolic disease, et cetera. And also perform the same phenotyping on what we called intermediate phenotypes, or people who have similar disease conditions, but without maybe overt pulmonary hypertension, with a goal of, it said here, deconstructing the traditional classification and defining new meaningful subclassifications of patients with pulmonary vascular disease. And I think we really have come to see that we may be able to refine the current clinical classification system to the point where we can use omics to help us better pinpoint what kind of pulmonary hypertension somebody has, instead of the approach that we presently use that's mostly based on clinical findings. So you can see the centers that were funded here. And again, the Pulmonary Hypertension Association generously funded one of the centers, which really made a huge difference in our ability to recruit in this trial. In fact, we completed enrollment right at the end of 2019. So you can see the timing was well placed so that we could do this before the pandemic. We have published in Circ Research in 2017 the goals of the study, and also a protocol for studying, for how a group of experts think that we could best study pulmonary vascular disease in humans. And I think this is a major contribution to our field, to have sort of a roadmap of best practices for studying pulmonary hypertension clinically in patients. And then we finally published our initial findings from PBD-omics in the Journal of the American College of Cardiology in the summer of 2022. And this is a schema of the main findings of the study. First, you can see the protocol that's on the left. So this was a really extensive protocol. All the patients had pulmonary function testing. They had a home sleep study. They had a CAT scan of their chest. They had a perfusion scan that was looking for blood clots. folks had echocardiograms, and then everybody with known or suspected pulmonary hypertension had a right heart catheterization. Obviously, the healthy controls that we enrolled did not have that done. You can see the final enrollment that's in the middle of the slide. It's predominantly group 1, but we do feel like we had a good representation of group 2 and 3 pulmonary hypertension. And excitingly, there are disease comparators here. And then there are novel observations from this cohort. Probably the most important one that my take home from this is that mixed etiology of pulmonary hypertension is common. So although we say a person has pulmonary arterial hypertension, it's not uncommon to also say that they may have sleep apnea and a component of group 3 pulmonary hypertension. And when I say common, it's actually about 40% of people after review by experts across the country. These experts could not narrow it down to one cause of pulmonary hypertension in 40% of the cases. During the course of our study, the definition of pulmonary hypertension was changed. So in the past, it had been greater than equal to 25 millimeters of mercury. But in 2019, that was changed so that pulmonary hypertension was defined as just greater than 20 millimeters of mercury of pulmonary artery pressure. And we had already enrolled a cohort of folks that were in this mild category 21 to 24 millimeters of mercury. And that's going to be a really exciting group to study. The last things that I was going to mention here that I think is interesting is shown in this spider plot. So I think one of the really exciting things about PVD-omics is to sort of challenge some of the dogma that we've had for years about pulmonary hypertension. And there are two things on this slide that I think are worth saying. One of them is that I was always taught when I was training that patients with pulmonary arterial hypertension should have normal chest CTs and should not have any what we would call infiltrates or anything different on their chest CT other than large pulmonary arteries. And these spider plots are done by group. So group 1, group 2, group 3, 4, 5 are shown here. And the percentage of people with ground glass or infiltrates on their chest CT is shown by the gradation on this internal line. And the interesting thing here is that almost half of the patients with group 1 pulmonary hypertension actually did have findings consistent with ground glass or infiltrates on their chest CT. So I think my takeaway was that actually this dogma that we've been taught for years is potentially wrong. And the other thing that I learned through seeing the data from this study is that I had previously always thought that patients with group 1 pulmonary hypertension or PAH were the only kind of pulmonary hypertension that should have a really reduced what's called DLCO. It's a marker of the ability of the lungs to take up air and oxygen. And it turns out that we learned from PVD-omics that that's actually not true. That about as many people in this orange line in group 1, group 2, 3, not so much 4, but 5 had reduced DLCO. So that dogma that I'd previously been taught that low DLCO is a sign of PAH is probably not true based on these data. And then the other major thing that we learned is shown here in the panel C, I think best in the upper right. And we looked at folks who had unfortunately had death or transplant after enrollment in PVD-omics. And we learned from this that the folks who had group 1 pulmonary hypertension, which are in green, tend to actually, so on this graph, it's percent without event. So it's better to be in the sort of lower parts of here. So pulmonary arterial hypertension in light green actually did relatively well, whereas the poorest survival was in group 3 pulmonary hypertension, where I would argue that we have very little therapies available to patients. And so I think the take home here was that a future area of focus should be in group 3 pulmonary hypertension to understand and treat this disease better. Interestingly, at ATS this year, Michaela Aldred, who's one of the investigators that's funded through PVD-omics, used genomic data to understand whether we might find genes that are associated with PAH in other kinds of pulmonary hypertension, like group 2 or 3. And she found that 3 patients who had COPD actually had gene variants that are associated with PAH, which was we would have anticipated they would not. and 3 patients with interstitial lung disease, again, a group 3 kind of pulmonary hypertension, had mutations that are known to cause PAH. So that was a really interesting and early finding coming out of our genomic data. And then the last sort of little bit that I wanted to share with you from PVDomics is using what's called a cluster analysis. So if you're not familiar with a cluster analysis, it is the task of grouping a set of objects in such a way that the objects in the same group or cluster are more similar to each other than they are to other clusters. And so one nice pictorial that I stole off the internet about how to do this includes giving yourself like what kind of person you were in high school. And I was somebody who was kind of out here on the grades, but on the athletics, I would 100% give myself a -4. So I would probably be really deep into the nerd territory here on this cluster. While my husband, who was great on the grades also, but like super athletic, would be gray here with the other people. Oops. And so he would be in the superstar category, I think, based on this, or maybe iconoclast. Either way, he was in a much different category. So these are different clusters that different people belong to, and this is just one way of clustering people. But it's using patients' specific characteristics to find who they're most similar to. And some really nice work from Andrew Sweat at Stanford had used cytokines or immune proteins in our body to define immune phenotypes in PAH. And he was able to use a cluster analysis like I just showed you to find that there are 4 distinct clusters of immune proteins in the blood in PAH patients, and these correlate with mortality. So that was a pretty interesting and novel finding when he published it. And it led directly into this paper that was published in Nature Communications in 2021, looking at RNA or transcriptomic profiles in idiopathic PAH and showing that there are 3 distinct subgroups of PAH based on RNA expression. So not just immune expressions in these, but any kind of RNA. And these also correlated with mortality. We are presently in the midst of doing similar analyses with the PVDomics data and excited to see more of that come out in the upcoming months and years. So getting to novel therapies in PAH, I just would like to address the elephant in the room, which is sotatercept in pulmonary arterial hypertension. So this is a drug that we talked about BMPR2 earlier, which is dysfunctional in PAH. But there's other receptors that are similar to that, including this ALK receptor over here. And we think that in the context of PAH, there's too much signaling through the ALK TGF receptor over here. And so sotatercept binds up the molecules that stimulate ALK in the hopes that you may have more signaling through BMPR2 pathway. And excuse me, there was a phase 3 trial in PAH. It enrolled people who were already on background therapy, which is a really important point. And found that, as published, it improved 6-minute walk distance. It improved 8 of 9 secondary endpoints, including quality of life, which I think is really exciting. And it's anticipated to have FDA approval in 2024 sometime. The other drug that's in a completely new class is seralutinib. This is a tyrosine kinase inhibitor that targets a number of different tyrosine kinases. It's an inhaled drug. It has completed a phase 3 trial where it met its primary endpoint of decrease in pulmonary vascular resistance. There were no safety concerns noted, and it was well tolerated. And presently, a phase 3 trial is being planned. So I think there's more to come on that. And I wanted to talk about an area of interest that I've had for a long time, which is physical activity as a therapy for PAH. It certainly is not going to be a cure for the pulmonary vascular disease, but it does offer hope for improving the lives of patients with PAH. And this builds on some early work by Meredith Pugh, who is a pulmonary hypertension provider at our center. And Meredith measured activity using an actigraph, which is kind of like a fancy Fitbit, in 20 patients with PAH and 30 matched controls, and found that there was reduced, low, moderate, and essentially zero vigorous physical activity in patients with PAH. And then if you look at the time of day and sedentary activities in PH, it's much higher than it is in control individuals. So based on that, Evan Britton and I designed and completed a mobile health intervention in patients with PH, recognizing that not everybody can go to pulmonary rehab and doesn't necessarily have the time, but we could perhaps intervene using texts and encouragement to walk more. And so what happened in this study that enrolled essentially any kind of PAH, everybody was given a Fitbit, and we observed them for 2 weeks. And then there was a baseline visit, and they were randomized to a texting intervention or to just usual, try to do as much activity as you can. And then they came in for a 12-week visit. And the primary endpoint was the step counts at week 12. So was this text intervention able to increase step counts in patients? And we did see with the intervention an increase in almost 1,400 more steps per day, whereas the controls over time had a slight decrease in steps per day. And you can see that shown here pictorially. The step counts in the treatment group are in green, and they increase substantially. The intervention arm took more steps on every day between day 9 and day 84 of the study, which was really an exciting proof of concept. Moreover, it did appear to improve quality of life in patients, and I think more work is needed presently to understand exercise as a potential therapeutic intervention in PAH. So there's a lot of other drugs, though, presently in the pipeline. Inhaled imatinib is one that I think holds a lot of promise. There's been interest in glycolysis with dichloroacetate as one drug targeting that pathway. Bartoloxone has been studied to enhance mitochondrial respiration. Fatty acid oxidation has been shown to be depressed in PH, and there's interest in drugs like metformin to increase that. And then recently, the GLP-1 receptor antagonists and also SGLT2 inhibitors appear to be interesting. And maybe actually, as shown here on the right, a little bit more at targeting the right ventricle, where we would also add fatty acid oxidation as a potential target also with the drugs that you see there. So I think this is an exciting time for research in pulmonary arterial hypertension, and I really appreciate your time in listening to this. I'm excited about the future, and I'm also really enthusiastic to hear what Julie Feitner has to say about her own experiences in this. So with that, I'll hand it over to her.
Thank you so much, Dr. Hemnes. It's always exciting to me as a patient to hear the research that's happening behind the scenes. So often as patients, our days are spent just struggling to breathe and survive. And to know that there's people whose daily life is spent behind the scenes trying to make that easier and try to prolong our life just helps give so much encouragement and strength for each day. So thank you all so much for the work that you're doing behind the scenes. As I mentioned at the beginning, I was diagnosed in 2008, and then the next day our son was born while I was intubated. I have idiopathic PH, and for the next 2 years, I worked my way through the medications that were available at the time. And at the end of those 2 years, I had gotten better, but PH is progressive, and I was already sick. And so I had reached the point in 2010 where it was either time for an immediate lung transplant and hoping I would survive the procedure because I was iffy, or there was a clinical drug trial that was opening up for imatinib. And my doctor said perhaps I might be qualified to join it, and I was. For me, imatinib happened to change my life, but I was at a point of being willing to participate in a drug trial because I was in a place of fear and uncertainty in my life. It was that or an outcome that I was not looking forward to as a young mom with a 3-year-old and a newborn. And as I went through the clinical trial process, I learned that it wasn't something I needed to be afraid of, but instead that my nerves were just because it was an uncertain unknown. And as I went through the process, it was actually an exciting process, an empowering process for me. Two of the first things that I learned going through the process were that as PH patients, we stay on the medications we're already on, that you don't, those aren't taken away from you. You want to do well, your doctors want you to do well, and so do the researchers. And so I'd at least be able to maintain what I had. And the second one was that if there was a hiccup and I wasn't doing well, I was free to step out of the trial. And so that gave me a sense of empowerment as I was willing to walk down this road. And both of those gave me a sense of peace as well. For the process itself, there's lots of testing that's involved. You want to stay safe and doing well, and so do your clinicians and the researchers. And so before starting a trial, there's plenty of testing that happens. Each trial is a different amount of testing, different testing that's done. And that starts you off as your baseline so they can see how you progress throughout the trial. And each trial, you go back into the doctor's office for visits so that they can see how you're doing, mark down any side effects that are going on, a different amount of visits, different amounts of time you'll be spending there. The trial pays for all of that. It's not coming out of your own copays with your insurance, which was a huge help for me and my family. The fact that patients never have to pay for that part. And I was able to, you could see on the slide that was up momentarily ago, that I was able to form good friendships with my research nurse to the level of even equal to that to my PH team themselves. And it's still become a lifelong friend. And I'm a person that I can still lean on today with my journey. So I was with imatinib for several years, and unfortunately, the FDA decided not to approve imatinib, but the science was good on it, the data was good on it, and so another company has been started up that was discussed earlier by Dr. Hemnes to try to get an inhaled version to market rather than the pill version with hopefully less side effects. And so that is exciting and awesome coming down the pipeline. But my experience with that drug trial was positive enough that when the opportunity came about a couple years ago for me to participate in another drug trial, there was not any fear or hesitation on my part again. This was for the sotatercept that's now hopefully going to be approved next year. Instead, it was just excitement when I found out that it might be a possibility. I was like, sign me up. How do I do the testing? I am all about this. The fear and hesitation had all gone because I had walked through the process once. And so it cemented in my mind that drug trials are not something to be afraid of for patients. They're actually empowering for patients. So if this is something that might be right for you, how do you find out about them? If you are at a larger PH center, your doctor may be participating in drug trials. If they haven't mentioned any to you, you could ask your doctor or your nurse about if there's any that are going on at their center for which you might be a possibility, and then go through the process of testing to see if you meet the criteria. The testing is for a baseline, yes, but they also have them nailed down in the trials to fit certain criteria of patients that based on their lab data, they think it'll work the best in or definitely work in. And so that's part of that testing process to see if you qualify for that part. It doesn't mean that down the road, if it's approved, it might not help you, but for the trial, they think that you might be one of the best candidates, at least to start with it. If you're not at an office, though, that participates in drug trials, doesn't mean that you can't still be part of them. You can Google for yourself what other trials are out there, or you can go to clinicaltrials.gov. It lists all the trials that are happening around the country, and you can filter it for PH or PAH and see what trials exist. If none of them happen to be at your center, then there's always a contact information there, and you're welcome to reach out to the researchers who are doing the trial. And sometimes they can get you into a center closer to you that is doing it, and you can still participate that way. And so if you don't happen to be at a large care center, a smaller hospital, or a smaller doctor's office, it doesn't necessarily exclude you. For the sotatercept trial I am currently in, my doctor's office is almost 4 hours away from me across state. And so the concept of this being a financial burden was something that was also weighing on my mind originally. But a lot of trials are all set up slightly differently. But sometimes they even provide things like reimbursement for transportation costs and things like that to help make that part not be a burden to patients. It takes my time, of course, to be part of it, but I'm also the one reaping the benefit of the drugs. And so if this isn't something offered where you are, still, I encourage you to see what else is out there. But let's say you're not quite ready to take the step and jump into a drug trial. There's still lots of other ways that you can be involved in research and help advance the knowledge of pH for yourself, for your friends, and even for people coming down the road, future patients. The PHA room at the conference that happens every other year is a great place to be able to jump in and be part of research. I've done that at each conference I've had an opportunity to go to. Sometimes it was a questionnaire I filled out or some blood work. I think one time they took like a mouth swab to get some DNA or something. And so it's all very easy things that you can do and still be part of it. Or sometimes when I'm getting a heart cath done or just going into my doctor for a routine visit, someone comes to me and asks, would I be willing to be part of a trial that day? That's not medicine, but just other things. Some things that I have done have been filling out questionnaires there. When I'm getting a heart cath done, on occasion, they've taken an extra vial of blood while they're already in there so that they could do research on that blood at that point and be part of a trial. And so there's lots of ways you can still participate in research, even if it's not necessarily the drug itself. And another way that I have been able to participate through the years is by fundraising toward research. In 2014, I started a 5K in Pittsburgh, and the first year the funds went to the PHA toward research. Some of the grants that they discussed today, events can help fund toward the research, or there's other organizations as well. But it was a way that I was able to do something. My friends were able to do something to help support me, help support other patients. And so there's lots of ways that we can participate as patients for ourselves. And so the point is that I have learned that research as a patient is very empowering. I can take pH by two hands, by two horns. One is by doing the treatments that the doctors prescribed to me on time, the way they said to do, and being honest with my doctor about when I'm not feeling as well, when I think things aren't going the best, so they can make tweaks. But the second one is to begin to do something about the disease itself by helping with research, helping with trials, helping to fundraise, helping to raise awareness. And so as a patient, so much is taken away from us, but by participating in the ways that we can, even from our couch, participating in the ways that we can to help bring awareness and help research to happen is just an empowering way to bring purpose from some of the pain that we deal with every day. And then the final thing for me to share before we head it back to Dr. Hemnes is that oftentimes as I'm speaking with patients about the concept of trials, they say that they don't want to be a lab rat. They don't want to just be a guinea pig for someone else to do science on. They don't want purple spots to appear or things like that. But I found that for myself and my experience, that is not how anyone treated me or looked at me or responded to me through the process. And going from science in theory to science research to a possible drug to FDA approval takes a very long time. And as patients, if we can step into the process as part of a trial, then it jumpstarts it for us. For example, I was in the original imatinib back in 2010. I'm still on the drug, even though it's not currently FDA approved, and now they're still working toward getting it approved. I've been on it for 13 years, where if I had just waited until this medication came out, there would have been 13 years of my life. I wouldn't have had the benefit of this drug. And this is true for every drug that goes to market. All of it has to be on trials ahead of time. And so by being willing to jump in and try to be part of a trial, it's helping people down the road, it's helping further science, but it can also be helping you today. And so I would encourage anyone, if they're able to, if they're willing to talk to their doctor about the possibility. And if there's any questions from a patient perspective, I'm always available. I'm on social media. I'd be happy to support you in any way I can. Thank you so much.
You, Julia. We review the questions in the chat box, maybe.
Hi, it's Angela. There are some questions that have come in. In the Q&A, there was one question that also came up that said, can we mix the medication we already take with new meds? I'm not sure if that's relevant for here, if you want to answer that.
That's a, a great question. It's really important to look at how people were allowed to enroll in trials of new medications in PAH. Most of the ones that have been done recently allow what we call background therapy. So endothelin receptor antagonists like macitentan, ambrisentan, or tadalafil, sildenafil would be okay. Sometimes they did allow folks who were using prostacyclins, even epoprostenol or treprostinil, to enroll. So most of the drugs that are being trialed moving forward allow these types of background therapies because they're completely new classes of medications and affect different pathways. So it would have to be specific to the drug, and your provider would have to look at, whether how that trial was set up. But most of the time, yes, they probably will be in addition to, not instead of.
There was another question. Isn't tryptophan in turkey?
I think so, but I can't say that we're planning any dietary modification trials that involve turkey. But that is a very good observation.
The next question I'm seeing here, there was some information shared about new potential drugs and treatments on the horizon. What is the data showing for these new drugs? Are there near-term trials associated with them?
So I'm not aware of any ongoing trials of dichloroacetate. There was one published in, I think it was Science Translational Medicine a couple of years ago. The trial of metformin that we ran at Vanderbilt is complete, and we're in the midst of analyzing our results and should publish it soon. So I think a lot of the ones that I showed as like potential avenues moving forward have either recently completed or should be completing soon. Probably the best place to find information about things like that are to go to the clinicaltrials.gov website. And you can search pulmonary hypertension and any of the drugs mentioned, and you can see what the state is of trials. We are required to update trial status, like enrolling, completed, etc. on there. So, and you can also see where you can enroll on that website also.
So I know we're right at time there. I have one last question I would like to ask everyone here on the panel, our HCPs on the panel. Where do you each predict will be the state of treatment options in the next 5 years? Or do you have a prediction? Don't all answer at once.
But I can go ahead and get started. So I think within the next 5 years, I mean, as Dr. Hemnes said, I think we'll see sotatercept roll out, and I think we'll get comfortable with using that drug and figure out the best way to use that drug. I think maybe on a slightly longer time horizon, we may start to see what I hope we'll start to see, which is more precision treatment. So understanding more about the specific molecular basis of disease in unique groups of patients, and match those groups to unique treatments in a personalized medicine kind of way. And my prediction for what will be the first drugs that roll out that fit that bill, my prediction would be that those would be metabolically targeted therapies, some of the work that Anne is doing with metformin, and then perhaps some of the GLP-1 and SGLT2 drugs that are out there for other disease states. But I do hope that maybe within the next 10 years, we'll start to see some personalized treatments come out.
I think that's a big thing in the next 5 years as well.
Thank you.
And I think the big challenge we're going to run into is especially with sotatercept hopefully coming soon, is how to combine all these medications, what's the right combination, when's the right time to start intravenous therapy now that there's other therapies that are available. So I think, although there might not be a lot more treatments available in the next 5 years, I think understanding the different treatment options and how to use them individualized in certain patients will be the next, I think, phase of this journey.
I don't think I have anything to add. I'm excited about all the same things, and I really hope that the future is brighter than what it is today. And even though we've made tremendous progress, I think there's a lot more room to grow, and I think we're chipping away at it. I think you're muted, Angela.
Sorry about that. I want to thank everyone again for joining us this afternoon. Thank you to our sponsors, and thank you to FCHP for partnering with us to offer this two-part series, which this is a reminder, we have the second part of this series on December 14th at 2 p.m., which will take a deeper dive into the clinical trials that we have available. And last but not least, thank you so much to our panel of experts, Dr. Hemnes, Dr. Simpson, Dr. Lammi, and Julia. We really appreciate you sharing your story with us. Again, thank you for joining. I hope everyone have a great day. And don't forget to complete the survey at the end of the webinar. It should pop up in the new browser. Thank you. Bye.